topic these days. Well, it probably was the hottest topic in science before this COVID thing came around. So it's kind of been pushed off the top space here of excitement in science. I'm not going to talk to you kind of directly about my own research at the moment. What I want to kind of discuss is the opportunities in this field for translating what we know into clinical practice, because I guess that's what everyone listening is interested in, translational medicine. How do we form those bridges between scientific discovery and clinical practice, coming up with new treatments? So I think Brian's already beat me to the spot for my first slide. I was asked to kind of give a little introduction to my career for people, so I won't dwell on this for too long. But as he said, yeah, I conducted I was the first year in UCD to do this course, human nutrition. And as you can see in that photo, I was the only guy in the class. It was a very female-oriented field, but I loved it because it allowed me to kind of join my love of food with science. I was very much into biology in school and figuring out how the body works. And I loved food as well. I come from a very kind of foodie family. And this allowed me to kind of study the basics of that, of how nutrients interact with our body to either prevent disease or in some cases to heighten risk of disease. So that was a great experience to have. And that during that time, I spent a year in a lab in Boston where I studied the interaction between nutrition and genes. So how some people with certain genotypes or carrying certain genes may be more susceptible to the effects of certain nutrients, which may have downstream implications for their health. So following this, as Brian said, I went down to Cork then and had a brilliant 4 years doing a PhD down in Cork. And I was kind of lucky at the point that I came down. The APC Microbiome Institute were just kind of taking off as a global superpower in their field. And studying all the types of microbes in the human gut and how that affected our health in many ways. So I was lucky that I kind of jumped on that bandwagon during my PhD and managed to start looking at how nutrients affected the gut and how that subsequently affected our health in many ways. But I was a dub in Cork, so I quickly found out that this is the case in Ireland, that you have Cork and you have the rest of Ireland when you're living down there. So I had to subtly hide my Dublin accent for my time down there. But I had a brilliant time both up in Fermoy and Teagasc and down in Cork as well, conducting a lot of this kind of mechanistic research into how nutrients affect our body and looking at the different pathways that are involved. And during that time as well, I was really lucky to be able to spend a year of my PhD back over in the US in Boston. And this again kind of provided me with another kind of perspective on how science works. And I really kind of recommend that to all the students listening, that you can get a completely different view of science and just a working environment by spending some time abroad, or at least in just a different situation that you're not comfortable in, which is good, and that you're not used to, because you can learn so much. That really has stood to me in my relatively short career so far. But over the last few years, I've been looking at a bit more translational approach, I suppose. During my PhD, I was looking at how certain nutrients affected our gut and our gut microbiomes. But now I've kind of applied this to a very specific disease context. And so we do a lot of work in Zimbabwe and Zambia, looking at kids who are either chronically malnourished or severely malnourished. And this kind of issue has kind of been forgotten about in recent years because there's a huge obesity epidemic, and we tend to look at overnutrition instead of undernutrition. But there's still about 1 million children every year die of undernutrition and its related problems. So my research looks at how the gut microbes of these kids may be contributing to their faltered growth or contributing to their death in extreme circumstances. And this again has allowed me to travel all over the world and spend time in different labs conducting some of the science to do this. And it's been fantastic. So I'm not going to talk, as I said, specifically about what I do, but I want to give you a little introduction to what the microbiome is. A lot of people might know already, but specifically how we can now move beyond just saying that we have this great ecosystem inside of us to translating that to the clinic or to the hospital. So if we picture an ecosystem, you might think of a rainforest or a coral reef like this here, like in Australia, the Great Barrier Reef. And the overall health of this ecosystem doesn't rely just on one species like the fish here or the coral or the microalgae, but relies on this constant interaction between species. And there's a food chain through which these different species interact and feed off of each other. And the entire health of this ecosystem really relies on this interconnectedness. And we've seen that with certain environmental stresses, like we see with global warming, that can trigger changes in very fragile ecosystems to really negatively impact the health of the ecosystem. So I'd like you to kind of think of that in the context of what I'll be talking about now, which is the ecosystems within inside of us, which is our microbiomes. So we are walking, talking ecosystems ourselves, covered in bacteria and viruses and a huge number of other exciting organisms. And the only reason that we haven't really been excited about them in the past is because we can't see them with the naked eye. And I think if we could see these things with the naked eye, like we can do here with a scanning electron microscope, we can see the diversity and the amazing nature that is not able for us to see with our current vision. So this is a scanning electron microscope of a stool sample from a patient, and it shows all the bacteria here in the gut feeding on a piece of fiber. So a lot of you might know kind of some of these statistics that are thrown around, and I won't dwell on them too much, but if we were to count up all of just the bacteria in our body, they would outnumber the number of mammalian cells, human cells that we have. These are very rough estimates, but we know that we have at least the same number of bacterial cells in the body as we do human cells, which is incredible to think the potential for that to affect our health. And if we're to look genomically, we know that there's 23,000 genes in the human genome, but we have well over 1 million microbial genes that have been identified. And so there is huge potential for these organisms to interact with all the different processes inside of us. But bear in mind that although we talk a lot about bacteria when we talk about the gut microbiome, really there's a huge number of other species which are completely unexplored and relatively unidentified so far, which arguably play a bigger role in the gut microbiome, but they just haven't been studied as much so far. And these include fungi and viruses and archaea and a huge variety of organisms. So we know what I've presented to you so far, that we have a diversity of organisms all over us, predominantly inside our lower gastrointestinal tract. But we also know that there are a huge number of unknowns, very much like the universe. There's been great science done over the years to identify stars and planets miles and miles away. But we also know that there's a huge number of planets or stars beyond what we can see. And so these are our known unknowns. And we have a similar thing in the gut microbiome. We have a huge number of known unknowns, despite what we've discovered so far. We know that up to 1/3 of the bacterial species in our gut are yet unidentified, which is fascinating to think that what we've discovered so far about the gut microbiome and what it can do to our health is only about 2/3 of the potential of what it could do. If we look at the genes, about 40% of the genes in our intestines from bacteria have no known function. They can't be annotated to any database to tell us what they do. And as I mentioned before, we've predominantly focused on bacteria and not viruses, parasites, and fungi. And COVID shows us just what can happen with one unknown virus. And imagine what other unexplored viruses are living inside of us and the potential benefits or risks that they could have. We also don't know what is a healthy microbiome. And that is one of the main questions on researchers' lips in this field, because we all have our own unique gut microbiome, which is as unique as our own fingerprint. And therefore, one person's healthy microbiome might look very different to another person's healthy microbiome. So we can't really translate this like other clinical measures, like a healthy blood sugar or a healthy cholesterol. And we can't have a kind of global marker of what is a healthy microbiome. And there are a huge number of other questions, like how do probiotics work? Probiotics have gained huge attention, both in clinical practice, in science, and in industry. But for the large part, we still don't even know how they work. And so that is another known unknown. But when you have known unknowns, you also have unknown unknowns. And these are the things that you don't know that you don't know. And so if we take this analogy of the universe, there are things that we don't even know that we don't know out in the other side of the far reaches of the universe. And that is the same for our gut microbiomes as well. I predict that there'll be a huge number of exciting discoveries over the next few decades that we hadn't even considered before in this field, which could have massive implications for translational medicine. So with that kind of introduction in mind, I want to give you 4 of my visions for future opportunities in the space that you're all in, which is translational medicine. And what are these, how these opportunities link science to the clinic. And my kind of predictions, whether they happen or not, of how we will approach these. The first of this is testing. And I think this will be really important in the coming years. How we measure the microbiome is going to be critical for how we target it from a clinical approach. And for years, the only way we can study microorganisms in any environment is by growing them up in culture, as many of you might have done in microbiology labs. But now we have these really advanced DNA sequencing technologies. And that is the sole reason that this field has evolved over the last 10 or 15 years, because of great engineering advances in how we read the DNA of living things. And it started with the Human Genome Project in the 1990s, and now it's expanded into the microbiome field. So we can read the DNA of microorganisms in the gut without having to grow them in the lab, which is very, very hard to do for many organisms. And so this is kind of portrayed by the cost, and this is one of the reasons it's happened, because the cost of sequencing has declined humongously. Back in around the year 2000, around the turn of the millennium, it cost $100 million to sequence the human genome, and now it costs 1,000, much less actually than 1,000 these days. So you can see that's the reason that we've been able to explore this field of reading DNA much more easily and much more cheaply. So what I think is going to happen, and what is already happening, is that these sequencing technologies will evolve even further. At the moment, if you want to read the DNA of a stool sample to look at your microbiome, for example, you have to send it off to a lab with a big sequencing device which sits on a bench, and it's about the size of 2 or 3 computers. But a few companies now have started working and have developed these handheld sequencing devices. And this is one of them. It's called the MinION. And it is about the size of your thumb and plugs in like a USB into a laptop. And this device itself has been used in tracking the genetics of Ebola, for example, during the Ebola outbreak in Africa a few years ago. It's even been used on the International Space Station to show that we can read DNA and look at microbial species in outer space as well. So this same company have even developed a device which sticks into your mobile phone as well. So what I kind of predict are the opportunities in this space are for these to become more applicable in the home through, for example, the use of smart toilets. And so one of these devices could sit in your toilet and you could read your microbiome after every meal if you have certain gut problems. And this will make our whole approach to reading and understanding our gut microbiome way more approachable for everyone. rather than just those who have the money to send off their stool sample just because they're interested in it. And this will allow us to move past just science and interest in what's inside of us, but moving to how these can be related to treatments. And secondly, in terms of testing, stool is not an accurate reflection of what is happening inside our entire gut. And I predict that we'll have to move past that if we're going to really tackle intestinal issues like bowel cancer, like various other IBD, IBS, and various other conditions of the intestine. And this was a study that looked at various regions of the intestinal tract, from the stomach all the way down to the lower intestine, to look at the types of microorganisms that were living inside there, both in the lumen and in the mucosa, the kind of sticky region lining the intestines. And what it showed was that stool, as you see over on the right here, wasn't an accurate reflection of the microbial environment of all these different sections of the gut. There were a huge number of different organisms living in the different niches of our long intestinal tract. And many of these intestinal conditions that we are focused on in this field occur in the upper intestine, for example. And so our stool microbiome mightn't accurately reflect what is happening there. So in the kind of sampling space, what I predict happening is that we'll move beyond just sampling stool samples, which have just been used because they're relatively easy to collect, and move beyond that to things like smart pills, which are already being used to a certain extent in clinical practice, where we may be able to swallow one of these pills and it can sample based on pH or based on a camera, knowing where it is in the long intestinal tract, and take readings and samples from various different regions to understand what is going on in upper regions rather than just in the lower gastrointestinal tract. There's some really interesting research also showing that because a huge number of bacterial metabolites are passed from the gut into the blood, that actually blood samples can give us a relatively accurate description of at least the diversity of the gut microbiome. And so there's potential there for finger prick tests, just like in diabetes, to be developed in the future to tell us a little bit about what is being produced in the gut and what has been passed from the gut into the bloodstream, which could give an indication of how leaky your gut is, which is an important phenomenon in things like IBS and IBD. Urine samples can also tell us a similar thing, but I think a real development in the space might be in breath samples as well. Because of the diversity of the organisms living inside of us, they produce a massive amount of volatile compounds and predominantly gases that we currently can't measure very accurately. But there is a huge potential for these to be read in some way, potentially through our breath, through things like developments in breathalyzer technologies. And I really see this as a potential for testing to be moved on in the microbiome field. So my second vision or opportunity in translational medicine relating to the microbiome is moving beyond what we currently know about probiotics. And probiotics has turned into a bit of a dirty word in this field, mainly because EFSA, the European Food Safety Authority, currently say that you can't use the word because it is technically a health claim, and no company currently has good enough science to call something a probiotic in a food, at least. Because it suggests that it has a health benefit that doesn't have rigorous enough science behind it yet. And so the evidence for probiotics is very, very mixed. And that's for a few different reasons, mainly because not enough good large studies have been conducted, but also because probiotics encompass a huge variety of species. And we can't lump the word probiotic, lump all bacterial species or all microbial species into that one term, probiotic, because there are such a diversity of species and strains that can affect our health, that they can all work in different ways. But we have hints of certain probiotics that do have potential health benefits, and this is one that was developed in Cork a few years ago now, and is one of the few probiotics on the shelves which does have good clinical evidence behind it. And this showed in a few hundred people, mainly in women, that taking this probiotic for 4 weeks could extremely reduce the symptoms of IBS compared to a placebo. So it was very effective. Things like necrotizing enterocolitis also have good evidence for being targeted by probiotics. So necrotizing enterocolitis is an awful inflammatory condition that predominantly occurs in preterm babies, and it causes inflammation of the intestine, can even contribute to death. And so there is quite good evidence of certain probiotics reducing the risk of necrotizing enterocolitis in preterm babies. But again, it depends on the strain that you use. And so we really need larger studies, which is tracking the same bacteria and the same probiotics to see how they work on a global scale. So the potential I see for this is that we'll move beyond what we currently view as probiotics. And we'll see more next generation probiotics, for example. These could be genetically modified organisms, which can translocate to a certain region of the gut, for example, or even target tumors, for example. There's some interesting research showing that that is possible. There's some interesting research showing that probiotics mightn't necessarily have to be alive, which is a bit of an oxymoron to their name, to actually be functional. And the sugars on the outside of their cell can actually trigger whatever responses you're looking for in the gut. And I think we'll move beyond bacteria as well, as I mentioned before, to start targeting, for example, things like prebiotics or these postbiotics, or all these fancy terms given to metabolites or fibers, which help to feed the gut microbiome in various ways. And one exciting...
Sorry, Rory. Actually, I think we're just running a bit late on time.
No worries, no worries.
Just, but as it happens, the question I was actually going to ask you was, funnily enough, about phage therapy.
Oh, there we go. Right on time then. Right on time.
Yeah, because I know that there's that's complete coincidence. There's a lot of work being done at UCC at the moment, I know, because of antibiotic overuse, and it just destroys all the good gut bacteria. So yeah, I'll actually leave it back to you, I think, just to answer that question, and then we might just and I'm sorry again, because I know all the stuff you've mentioned is extremely interesting, but
you're all right, don't worry.
In the interest of time, thank you.
You're all right, don't worry. So phage, as people know, are viruses that infect bacteria. And so, as you said, Brian, probably one of the biggest health challenges of our time after COVID is going to be antimicrobial resistance. And we're all the microbes that infect us around the world are going to become resistant to all these antibiotics that we've overused over the years. And so we'll need more targeted approaches. And so there's a huge potential for these phage, which you can see this kind of green alien-like structure here, to be used as treatments to specifically target certain bacteria, rather than using broad spectrum antibiotics, which kind of tend to kill everything in the environment. And it has been used in very rare circumstances in clinical practice already. There was a case in London about a year or two ago of a young girl who had an awful infection and was resistant to a lot of antibiotics and had this phage therapy, and it kept her alive, which was amazing. So I can see that as a potential therapy in the future that will be developed more. And as you said, a lot of that work has been done in UCC, and it's really exciting.
That's great. I just wanted to thank you again, Rory. Your talk was exceptional, and I know you only got to do 2 of the 4 points, but
all right, don't worry. I'll save the other 2 till the next time, till next year maybe, and we'll leave it till then.
That would be great. Thanks very much. I think you just gave us an insight into just how unexplored this area is, and as you said, the amount of unknowns we have. It's such an unexplored frontier, but we're very fortunate here at UCC. We have such a developed world-class facility, so it's great to hear about it, and hopefully we'll see you again. And thanks a million for coming on and speaking for us today. No worries.
All the best for the rest of the day. See you later.
Thanks, Roy.
Thank you.
Bye. Thanks, man.
Thank you.